I think all have joined. Yeah. So, good morning, students. Let's begin today's class. And today, what we're going to do is we're going to continue our understanding on the chapter that is biological classification. And in that, if you remember, we are talking about the 5 kingdom classification system, and we have progressed. We have completed the kingdom. First kingdom that we talked about, that was kingdom Monera, and from there we will understand this next kingdom today, that is going to be kingdom Protista. So anybody can summarize what we have learned in the previous class regarding kingdom Monera? Any one person? Can anybody summarize what we have learned in the previous chapter? Because there are some correlation that we will make from here to here. That's why I'm asking. Anyone? Whatever you remember.
Sir, in the kingdom Monera, only prokaryotes are present.
Okay, very good. Only prokaryotic organisms are present. Prokaryotic, here carrion refers to nuclei, and the primitive nuclei, right? When you're going to have nucleoid, this type of organism, this type of cells, prokaryotic cells, which are unicellular or multicellular. Are they unicellular or multicellular?
Unicellular, sir.
Unicellular, sir.
Unicellular, sir.
Single cellular, sir.
Very good. So here we have to understand that it is unicellular and prokaryotic. So here, kingdom Monera, we said they are going to be unicellular and prokaryotic. Please switch on the microphone. Switch on the microphone, please. Yeah. And here, what we do, see, when we talk about the classification system, right, in the previous classes, and you should understand that when we talk about classification system, we always go from something simple to something complex. And here we have started one character, one of the traits that we talk about, and we classify the organism is going to be the organization of the cells. That is, whether they are unicellular or multicellular. So here there are organisms which are unicellular. So the next step in the process of classification, the next hierarchy should be what? Of the multicellular organisms. But we are having a second feature that we are a prime feature that we are talking about, and that is the prokaryotic organisms, prokaryotic cells. And we know the next evolution, the next higher group is going to be in this respect is going to be the eukaryotic organism. So when we talk about the next kingdom Protista, we are talking about eukaryon or eukaryotic organisms, eukaryotic cells, right? They are going to have a true nucleus. Here going to be U refers to, right? This refers to true, and carrion, right? Here this carrion word refers to the nuclei or nucleus, we can say. So they are going to have true nucleus, not nucleoid. They're going to have the nucleus where we're going to have the nuclear membrane that is going to enclose the nucleoplasm, and that nucleoplasm is going to contain the chromosomal substances, you can say genetic material, which can be the DNA typically, right? Double-stranded DNA typical cases. So here, another thing that you have to keep in mind, it is not multicellular, and typically here they will be again unicellular. So these are also unicellular, but what is the difference here? These are eukaryotic in nature, and that simple change in this prokaryon to eukaryon changes a lot of, makes a lot of difference when we go from this particular kingdom to the Protista kingdom. The organization, the way they behave, right? The nuclear organization, the cellular organizations, everything change when we change this simple word from prokaryotic to eukaryotic. And we understand that it is actually progressing, it is evolving. Right, they have evolved, they have evolved, and we're going from a simple to slightly more complex, slightly more organized, slightly more advanced. Right, and when we go to the next kingdom, in the next kingdom, we are going to have the same eukaryotic cells, because we know that it is only the Monera that is prokaryotic in nature, and the entire remaining kingdoms, they are all going to be your eukaryotic in nature. So here it is eukaryotic and unicellular. The next kingdom is going to be eukaryotic and multicellular. Right. So with that, let us start our understanding of this particular concept. I'm going to now share with you all the screen, the PowerPoint. Okay, I'm sorry, not this one. Okay, can you see the screen?
Yes, sir.
Okay,
Yes, sir.
Yes, sir.
Yes, sir.
good. 1 second, let me change this setting. Yeah, we're good to go. Kingdom Protista. Okay, now anyone can tell me what is this particular image that is right now shown here in this particular image here? It's an organism. Can you tell me what organism it is? If not, any, we'll come back to this. Okay. Yoglina, yep, that's correct. Anyways, let's move on. So here when we talk about protists, it is going to include all the unicellular eukaryotic organisms, right? And these particular kingdom, the protists or the protista kingdom, they're going to form a link between the next kingdom, that is going to be the kingdom fungi, and or the kingdom mycota we can call it, and the previous kingdom, that is going to be the kingdom monera. So because as we said here, they are going to have the in case of protista, they are going to have the unicellular eukaryotic organism, whereas when we go to the fungi, in the fungi or in the mycota, we are going to have the multicellular and eukaryotic organisms, right? And that is why this particular kingdom is going to be an important link, important phylogenetic link. Phylogeny, when we talk about the evolution, right? Phylogenetic link between these two kingdoms. And now they're also going to be consisting of the phytoplanktons. You have to understand this term. Now, what are phytoplanktons? The protists or the member of this group with chlorophyll and those organisms that are capable of photosynthesis. For example, diatoms, right? Diatoms is one type of protist which are going to have the photosynthetic ability because they contain the chlorophyll pigment, and this is why these are called the phytoplanktons, right? You would have also heard about another term, and that is called the zooplankton, right? These are zooplankton. These are kind of animals, and here we are going to have kind of not animals exactly. So here what we're going to have the phytoplanktons as major food producers in the ocean, right? They're typically present in the ocean, and because they are going to be they're going to be photosynthetic in nature, they are major producer in the ocean, and based on that, right, the other organisms are going to be dependent on the phytoplanktons for the food resources. Now, what is the structure of these organisms? As we said, these are eukaryotic in nature. So all the features that you know about eukaryotic cell holds good for these organisms as well, because they're unicellular in nature. So here the cells are going to have a true nucleus, and there is going to be nuclear membrane, of course, and there is going to be nucleoli. Now nucleoli is a structure, membrane less structure inside the nucleus. So there is a difference between nucleus and nucleoli. Nucleoli is going to be present inside, that is going to help in the production of certain RNA, certain ribosomal RNAs actually, which we will talk in the later part in the cell chapter. Now they are also going to have membrane bound organelles. Now if you go back, if you remember the kingdom monera, in kingdom monera there was no membrane bound organelles, right? And also there was no true nucleus. Now many in the previous case they all had the cell wall, right? Most of them had the cell wall except the mycoplasma in case of monerans. Now in case of protist, they also may or may not have the cell wall. It is not compulsory for every protist to have the cell wall. And when we talk about the photosynthetic form of these protists, they have chlorophyll, they have the chloroplast, but they do not have the cell wall, right? When the photosynthetic form, autotrophic forms in these do not have the cell wall, but do have the chloroplast. Now, how do they move, right? What is the locomotion mechanism? There are 5 different types. What is the first one? Flagella. Flagella and cilia, you might be knowing about it, right? The flagella is a long whip-like structure. Anybody knows what whip are? W-H-I-P, whip. Do you know what whip are? Anybody? Hello? Can you hear?
No, sir. No, sir.
Okay. In Chabuk, you know, in Hindi movies you would have seen.
Yes, sir. Yes, sir.
Yes.
Yes, sir.
Yes, sir.
In English we
Sir.
call that as the whip. So what happens when the let's say you want to control the animal, you rotate the entire weight and hit the organism, right? It is a complete one round structure. And the locomotion in the flagella is going to be something similar. So the entire flagella rotates, and that will basically propel the organism toward the front side, just like a whip, how it is going to move. And one cell typically have only one flagella in most of the cases, but in some cases it may have more than one also. Now coming to the next one is the cilia. Now cilia, you have to understand, these are much smaller. I'm sorry. I think it's, yeah, okay, so much less. Yes. So cilia, you can see they are much shorter, and they are going to work in a coordinated manner, because they are not going to be present in one or two. Instead, they are going to be present in thousands of the cilia are present all over the cells. Now, to make you understand that, how many of you have seen that boat rowing, a race in let's say Kerala boat race, long boat with 30-40 people will be sitting in the
Yes, sir. Yes, sir.
right? You have seen that?
Yes.
Now imagine the entire boat. Now imagine the entire boat to be the organism, right? Now what is going to happen? Both the sides of the boat, that is both the side of the organism, is going to have number of small bristle structures. That is your oars, right? And that is rotating always in a coordinated fashion. Now what will happen if, let's say, out of the 30 people, on the right side people, they start to push the boat forward, and on the left side they start to push it backward. So that will not work. So everybody, all 30 people, both on the left and right side of the boat, must row the boat in the same manner, in a coordinated manner, so that the boat moves either the front or the back. Similarly here, the cilia, which is actually similar to that small structure, present numerous in the organism, they're going to work in a coordinated fashion, so that they will actually move the organism in a particular direction. Yes, somebody has raised the hand. Anybody wants to ask a question? Anybody wants to ask a question? I guess no. Next is going to be pseudopodia, right? And that commonly we all know as the false feet. Now this is a term that I have seen many of the students even in class 11 they use. All right, but what you should try to understand is this is not the false feet actually. We use the word, but they are actually protoplasmic extension of the cell. Right? You have the entire cell, and some amount of the protoplasm is going to be extended towards a particular direction that looks that kind of relates to the feet. So that will allow the organism to move in that direction. For example, the protoplasm is extended in one particular direction, and then the organism is going to be pulled towards that particular direction, or that particular that spot. And after that, again there is going to be extension or the pseudopodia, and that will allow the organism to move. So you have the flagella, you have cilia, then you have the pseudopodia. The next one is going to be the wriggling. Wriggling is a mechanism, again in some of the protists. Here what happens is there is going to be twisting movement. Why this twisting movement? Because there is going to be contraction and relaxation, or contraction and expansion of the cells. The entire cell here is going to contract and relax, and that will basically push the organism in a particular direction. Next one is going to be the last one, that is mucilage propulsion. As the name suggests, here the organism is going to secrete mucilage, and it is going to move in one direction, right? Whichever direction the mucilage is going to be produced, the opposite direction the organism is going to move. A very vague connection to this is just like rockets. What happens in rockets? We are going to have the rocket move in one particular direction. If it is going from let's say towards the upside, what happens? The thrust or the fuel that is burned, and the the thrust is produced on the bottom side, and that thrust pushes the entire rocket towards the top. Similarly here also something similar happens, where the organisms like the diatoms, they are going to produce the mucilage, which is going to propel the organisms in the opposite direction. So when we talk about locomotion in the protist, you have to understand these 5 different types. Yes, anyone is having any question on this, or did you understand or not? Okay, so Pratima is asking, what does mucilage mean? You can use the microphone also, because every time I may not be able to see the chat. Okay, so please use the microphone. So what does mucilage mean?
Yes, sir.
That is Pratima's question. So mucilage is basically a slimy substance released by many of the organisms. Okay, and many times that is for the protection of the organism, uh, both in case of the plants and animals. Okay. Okay, so now we talked about the structure, we talked about how they move. Now the next point is what is a nutrition mode in case of protist. And now here you will see that this particular group, right? In the previous case, what we have seen that they are sorry, that they are autotrophs or they are heterotrophs, right? And when they are autotrophs, in case of Monera, I'm talking about. So when they are autotrophs, they can be chemosynthetic autotrophs or photosynthetic autotrophs, and they can be parasite also. Now, when we talk about the nutrition in protist, we have organisms that are photosynthetic, we have organisms that are following the holozoic mode of nutrition, then you have organisms that are following the saprophytic mode of nutrition, and then you have parasitic mode and mixotropic mode. This is very important. So here you see 5 different types of nutrition in the organism. Now, when we talk about photosynthetic, as the name suggests, here organisms have photosynthetic ability because of the presence of chlorophyll pigments, right? And here you can see the examples as diatoms, euglenoids, dinoflagellates. Again, we're going to talk about this in the types of the protists, so do not worry about the names right now. You have to remember, but at a later point of time. Now the second one, holozoic. Holozoic means what? The entire food is going to be taken in and it is going to be digested inside. For example, amoeba. What happens in amoeba? Amoeba is going to engulf the entire food. And inside the organism, inside the amoeba, you have learned in class 10, inside the amoeba, there is going to be a large number of these chemicals released, and this chemical digests the food material or the organism inside, and then assimilate that digested food material to produce energy. And whatever is extra, whatever is unnecessary, that is going to be thrown out of the amoeba body. I'm sorry. Next, we are going to have the saprophytic mode of nutrition. Now, the saprophytic, as the name suggests, they are going to be feeding on dead and decaying organic matter, as we discussed yesterday also, right? And one of the example of that is going to be the slime molds. Now, these are very interesting organisms. Slime molds, I have some video to show you. If I'm not able to show you now, I'm going to send you the link. And in my opinion, you must see that video. How unique these slime molds are and how different they are from the entire, the remaining of the living organisms. Very unique. I'm going to send you the link of the video, or if I'm going to get time, I'm going to play it here also. Now, next one is going to be parasitic, as the name suggests, very simple. They depend on the host organism, and the examples are Plasmodium. Right, malaria is caused by the Plasmodium vivax, Plasmodium falciparum. Then Trypanosoma brucei, they cause the sleeping sickness. Right, then you have Entamoeba causes the amoebic dysentery. Entamoeba histolytica is the name. Amoebic dysentery they cause. So these parasitic protists, they cause number of the human diseases that we all suffer from. And finally, the term mixotropic. Right, when we talk about mixotropic, we have to understand that mixotroph, right? Any idea what it could be? Based on name, can you tell me? Anyone, any guess what it could be? Mixotroph. Guess, any wild guess?
Sir, it is a mixture of the.
Yes,
Sir, it is a mixture of the tropic movements.
See, we are talking about what? Nutrition, right? We are talking about nutrition. So in that you try to relate. Tropic
Sir,
movements.
it might be exotropic and autotropic.
Yep. So here the organism is going to exhibit both autotroph or autotrophic mode and heterotrophic mode. Right. We are going to have both the autotrophic mode and heterotrophic mode of nutrition that is going to be exhibited by one organism, and in that case we are going to call a mixotropic mode of nutrition. Right. So here an organism that is living in certain condition, it might act as a autotrophic organism because it will contain chloroplast. It will contain chlorophyll. Exactly, chlorophyll pigments will be there. But now, if the situation is not conducive, the situation is not proper for the food to be produced, for example, sunlight is unavailable. At that point of time, what they will do, they will give up the entire chlorophyll pigments. They will not use that, and they will rather enter the heterotrophic mode of nutrition, and they will start feeding on others. Now that is something unique, and you see that in case of Euglena. That is something, the reason why Euglena was able to place in any of the groups. Again, when we talk about in the detailed class later in the NEET, I will tell you why was Euglena not placed, was difficult to place in a particular group. So Euglena is going to be both autotrophic as well as heterotrophic mode of nutrition. They exhibit both. Now, when the sunlight is available, when the environment is all right, they can produce the food. At that point of time, they follow the autotrophic mode of nutrition. But when they do not have the sunlight, they shift to the heterotrophic mode of nutrition, and that is why it is called mixotroph or mixotrophic mode of nutrition. Are you clear on this?
Yes, sir.
Yes, sir.
Yeah. Okay. Good.
Yes, sir.
Yes, sir.
Shall we move on?
Sir.
Yes.
Okay. Very good. Very good. Now, reproduction in protists. So the next component we talk about how the reproduce, how these organisms are going to reproduce, whether it is sexual or asexual. So here you have to understand that the reproduction in protists is going to be both asexual and sexual. Now, what you have to keep in mind when we talk about the monera, in monerans, the reproduction was only one type, binary fission reproduction, just cell division, asexual mode. There was no sexual mode of reproduction that we know of in case of monerans. But in protists, it is the first time that we see an organism reproducing by the sexual mode. Again, this is a new feature that has actually been added in the evolution process. And when we talk about the asexual mode, what are the different types? We have the fission, we have the plasmatomy, and we have the cyst formation. Now, all of these we will talk about in detail at a later point of time. Now, along with that, in the sexual mode of reproduction, it is going to be with the help of the meiosis. Again, it's a very, very important factor, very important, evolution mechanism. I'll tell you why. See, always we know that there are going to be the germ cells, right? And germ cells are important for the sexual mode of reproduction. Now, the germ cells are going to be diploid in nature, and they are going to be formed from the somatic cell. What is somatic cell? Your regular body cells. And these somatic cells happens to be diploid in nature. And there will be 2N. And one second. Yeah, I hope you can see that now. So what happens is we are going to have the somatic cells or the regular body cells that we all are made up of. Any organism is made up of. Now, when we talk about sexual mode of reproduction, we are talking about the presence of specialized cells, the involvement of these unique specialized cells that are developed only during the reproduction process. And these are called the germ cells, and they are having half of the chromosome of the original cell. Now, if you all have a little bit memory of class 9 cell division chapter, cell chapter, at the end there was a concept on meiosis and mitosis. I hope you remember that. If not, try to understand now. So it is by the process of meiosis that we are getting this specialized germ cells. So if I take the examples of humans, the somatic cell in our case is going to be consisting of 23 pair or 46 chromosomes. But in the germ cell, it is only 23 chromosomes that are actually present. That means out of the 23 pairs, it becomes only 23. And that is possible because of meiosis. And meiosis in the evolutionary process, the introduction of this, or when we saw it for the first time, it is a very important factor, because in the higher organisms, we are not going to follow the asexual mode. It is only the sexual mode that is actually involved. Because sexual mode is supposed to be better. It is going to give us more variation. It is going to be more stable. It will allow for the evolution to go on. And that's why the nature is going to prefer sexual mode in the higher organisms rather than asexual mode. And that is the reason why we have to understand that. That is the reason why when we talk about the sexual mode and when we talk about the meiosis in the protist, we have to consider that as a very, very important phenomenon or important introduction when we talk about evolution in the organisms or in the animals. I hope you are clear on that. समझ में आ रहा है कि नहीं?
Yes, sir.
Okay, let's move
Yes.
on. Let's move
Yes, sir.
on. Okay. So now, how do we classify the protist? Right? What are the different groups that we can make out of protist? And again, when you see this, you will understand some of them. So when we are going to have the protist, typically we can group them into three categories. What are the three? As you can see on the screen, it is a photosynthetic protist. As we said, nutrition, it can be photosynthetic or it can be that is autotrophs or heterotrophs. So based on that, we're going to have photosynthetic protist or the algal protist, we call it. Next, we have the unique, as I said, the slime molds. And finally, we are going to have the protozoan protist, the protozoan protists, protozoa basically. And the first one, there are going to be three types. The algal protists or the photosynthetic ones that are going to be Chrysophytes, dinoflagellates, and euglenoids. Three categories. Again, all these three are new terms. Most of these are new terms. And my dear students, you have to somehow understand and remember it. A little bit difficult, I do understand that, and I told you that in the beginning itself. These terms are new, concepts are new, and there is hardly anything. If you are interested in this, very good. If you want to learn about the classification, diversity, well and good. But I have seen many people do not find interesting. But I personally do a lot of interesting. I do find it very interesting because if you go onto internet and you try to search the organisms which you see here, for example, type of Chrysophytes, dinoflagellates, you find something very unique. You find features, you find the phenomena that you typically do not see around, because their habitat is different and some of the mechanisms, some of the the processes are unique only to these organisms. So if you are interested in biology, in my opinion, you must see some of these video clips. Anyway, if you are interested, you can ask me. I will try to send you the links. Anyway, the first category now is the Chrysophytes, right? And these are called they are going to include the diatoms and the golden algae. And they're microscopic, eukaryotic, and uninucleate diploid cells. That is the regular features of the group. Now, the cell wall forms two overlapping shells which fit together in a soap box. What is the meaning of that? I'm going to show you that in the picture. Now, what you see, yes. Now, what you see here in this picture is this particular part. Okay, we are running out of time. This particular part is going to be one particular shell or one particular cover, and as you can see, this particular part, it is another covering. Okay, this is by the way a diatom in under electron microscope. Okay, and you can see this. It is the scale is 3 micrometer. It's very, very small actually. Now they're fitting together like a soap box. They're fixed here. You can see this center part is going to separate out both the organisms, and you can see even here. Now these are going to have a very highly ornamental structure or sculptured cell wall, and that's why they are referred to the jewels of the plant kingdom. Now as they are photosynthetic, they are the chief producers in the oceans. They produce the food. They are the primary producers. Now, as you can see here, this particular, the interesting structure that you see, the design you see, these are all of the cell wall design or cell wall structure. Sir, all the organisms kingdom producer aquatic. We'll come to that. Yes, marine. Most of them are marine, but again, few examples I will tell you which are not. So let me finish this part. Okay. So as you can see in this particular picture, you see this design, and that is because of the cell walls. That's why they are ornamental in nature, right? They are the jewels of the kingdom here. Now, next, let's go forward and understand the next one, that is called the Chrysophytes. Now, here the cell wall is going to be hydrated silica. They made about hydrated silica and another compound called pectin. It's a biomolecule, and with some of the cellulose. Now, they form or they're involved in formation of something called diatomaceous earth. So what is that? So the place where they live, where the Chrysophytes are living, right? They are going to leave behind a large amount of cell wall deposition, and this cell wall deposition, it is going to accumulate over billions of years, typically at the bottom of the ocean, and these are going to be called diatomaceous earth. Now, what benefit are they having? What work or what use they are having? They are going to be used in the polishing industry, filtration of oils, syrups, because of their gritty nature. They are not very fine, they are slightly coarse, and because of that, they are used in these different conditions. Now, there are two types of diatoms here you have to understand. One is called the pennant diatom, which are small rectangular soapboxes shape, and there are centric diatoms, which are going to be circular in shape. I think this part of the previous one I have just misplaced it here. Anyway, so you can see here some of the diatoms picture actually. Okay, you can see the unique sculpturing here. Okay, and this one is actually the diatomaceous earth. Right, actually it is available in the market. Right, it is actually marketed in the. It is available online also. Now the third one is called the dinoflagellates, and here they are also, most of them are marine, not all, most of them are marine, and they are photosynthetic, because all three chrysophyte, dinoflagellates, these all are your photosynthetic organisms, and here the cell wall contains cellulose, right, and they are going to have stiff outer cellulose plate, and most of them have two flagella. Right, as the name says, diano, means you are going to have di means two. So two flagella, that's why the name dinoflagellates. And one is going to be longitudinal and other is going to be transverse. As you can see here, this particular image you see is of the peridinium, and this is a motile dinoflagellate. And as you can see, there are this cellulose plate, as you can see here, right? The cellulose plate, and this one is a longitudinal flagella. And in this particular one here, this is the transverse flagella. So there are going to be two flagella, that's why the name dinoflagellates. This is an electron microscope image of the same dinoflagellate. Now they are going to have some of these dinoflagellates contain pigments. For example, they are going to have the different type of colored plastids, and these colored plastids give a unique color to the cells. Again, something this is new, and these pigments are called the chromatophores. Now, in some cases, what happens? These dinoflagellates they present in large population, and they quickly make the sea or the water or the habitat they're living in to look red. Now, what I'm talking about, you can see here, right? This particular part, it looks red, right? And that is because of this dinoflagellates. So they can quickly change the color of the water, and typically the toxins that are released by these dinoflagellates, they can kill the marine organisms, some of the other marine organisms or animals. So these are called the red tides. Now,
Sir,
yes sir, yes sir.
are they harmful to humans?
Yes, they will be harmful to humans. If something is harmful to animals, they will be harmful to animals also, humans also. But as I said, they are not going to be found in every single place, right? They are going to be localized, and typically this red tide occurs not always, but only at certain times. Typically when they are defensive, right? If all the time they are releasing the toxins, all the time you see the red tides, I think we may not have any other aquatic organisms, but that is not the case. So only at certain times they are going to have the property. Am I clear?
Excuse me, sir.
Yes, sir. Yes, sir.
Sir, why they have two flagella, sir? What are
Sir,
the uses?
okay. Again, they are going to have different mechanisms which are going to be beyond the scope of this class. When we talk about in the NEET classes, we are going to talk in details. I hope the classes start soon. Because there are many things that we have to discuss, but again, if you discuss that, we cannot move forward with this in CRT ones. We will talk about that. Do not worry about that. And again, the number of flagella, again, the main function in many of the organisms is for locomotion, and in certain cases for the trapping of the organisms, trapping of the prey or the food, for the food particles. Another factor, yes, good. Another factor that you have to keep in mind, we were talking about it yesterday, that is the bioluminescence. And some of these dinoflagellates, they are going to exhibit that. That is, there are going to be light that are going to be emitted by the dinoflagellates, and this one is actually simulated in a laboratory condition. Now, this particular bioreactor contains the dinoflagellates. As you can see, they are glowing actually in a completely dark condition, dark environment. So bioluminescence is the property of living organisms giving off light, and that also we see in this particular case. And the last one in this case is going to be the euglenoids, that is going to be euglena, and they are mostly freshwater. What you saw till now in the previous cases, marine, and now freshwater organism, that is euglenoids. They have a pellicle, that is going to be a protein-rich layer instead of cell wall. There is no cell wall. We said that they may or may not have a cell wall. So in this case, you do not see the cell wall. What instead you see is a pellicle, thick protein layer, and that makes the body flexible. And they are also going to have two flagella. One is long and one is short. The mode of nutrition that we have already said is going to be autotrophic and heterotrophic. And as you can see here, now this one is going to be the larger flagella and this is going to be the smaller flagella. And here you're going to have the chlorophyll pigments, the Golgi apparatus, mitochondria, and also you're going to have the nucleus. And this flagellum is going to help the organisms to move. So that's the microscopic view of the euglena that we talk about. And as you can see here also, you see the chlorophyll pigments and you can see even the flagellar structure. Right, one flagella you can see. So that's all regarding this concept on the first group of the kingdom protists, or the organisms of the protists that are going to contain the chlorophyll pigments. Tomorrow we'll talk about the slime molds with certain video that will be pretty interesting to see. Yes, anybody has anything? We have around 1 and 1.5 minutes. Anybody wants to ask anything? Yes, sir. Samiksha.
Sir,
Okay, yes, quickly, yes.
Sir, why one of the flagella is larger than the other one?
As I said, we'll be discussing that, not right now. One flagella, why there is going to be two flagella. Again, one more condition I'll tell. In one case, you'll get two nucleus also. Right? There's something called micronuclei and macronuclei. Right? Why these things are there? Why these unique things are there? We'll talk about that in a particular class. Do not worry about it. Samiksha, I will be explaining
Okay,
that.
sir.
Yes,
Okay,
I'll be explaining
sir.
that. Anybody else? Anybody else?
sir, doubt, sir.
Yes. Siddhi,
Sir,
yes.
bacteria structure is very simple and complex.
Yeah, you have a question. I got it. So there you have said the bacteria is supposed to be so simple, and why we call it complex, right? Mechanism is complex. Just to that, I will just give you one simple analogy. So the bacteria are very interesting because the moment we put, let's say, antibiotic,